Hello everyone. Welcome to the General Microbiology Lab Session 8-9. In this video, we will be discussing about microbial growth, and it will be delivered by me, Rafsan Shabani Khalid. So let's begin with the learning objectives of this laboratory. Okay, firstly, we would like to reintroduce basic microbiology techniques, particularly aseptic pipetting. And then the second one, we would like to observe the growth of bacteria in different growth stage. And lastly, we would like to plot a growth curve and to analyze the different phases of microbial growth with regards to intrinsic and extrinsic factors. So let's begin with the overview as well. Let's discuss a little bit about overview. Okay, the growth of microorganisms can be differentiated into four general phases. Okay, first one is log phase, and then exponential phase, stationary phase, and death phase. Okay, of course, during these general phases, there will be some factors that may influence these growth phases. Okay, it can be intrinsic or extrinsic factors, such as the growth media, optimal temperature, pH, and indigenous factors, for instance, metabolic pathway and genetic linkage. Right then, as it has been explained from the previous slide, there are several phases for the growth bacteria. Okay, and the first one is log phase. As you can read, there is no increase in number of living bacterial cells. Why? Because the bacteria adapt themselves to the growth conditions. It is the period where the individual bacteria are maturing and not yet able to divide. And after that, the next stage is the log phase. And they try to divide themselves, exponential increase in number of living bacterial cells. This is a period characterized by cell doubling. Okay, and then the third one is stationary phase. In the stationary phase, is often due to a growth limiting factor such as a depletion of an essential nutrient. So that's why it has a plate number of living bacterial cells. Okay, and the rate of cell division and death roughly equal. And the last one is the death phase. This is where the bacteria die. Okay, the exponential decrease in number of living bacterial cells due to the lack of nutrients. It can be environmental temperature above or below the tolerance band for the species or other injurious conditions. I hope this bacterial growth is quite easy to understand. So let's continue. Now let's discuss how the bacteria actually reproduce. The bacteria reproduce through a mechanism what we call it binary fission. Okay, as you can see from the picture that the binary fission, they are separated into several steps. Okay, the first one is DNA replication, and then the second one is the formation of division septum, where the cell try to elongate themselves, and then after that the cell separation. And by the end there will be two sister cells that they are actually the same as the parent. Okay, please remember that the binary fission because they try to divide themselves, and this can be expressed as 2 power n. So this is the formulation how we count the number of the bacteria. Okay, so let's imagine. So basically, the bacteria cells they are divided every 20 or 30 minutes. Let's say 30 minutes. Then in 24 hours, the amount of the bacteria would be 2 power 48, right? Because 1 hour is 60 minutes and they try to divide every single 30 minutes, right? So after 24 hours, we just multiply 24 hours by 2, it means 2 power 48. And the amount of bacteria itself would be 281 trillion. approximately. You can imagine if someone get exposed by harmful bacteria, they will just right after 1 day, 2 days, they will get sick because of this. The amount of bacteria and the mechanism they reproduce through binary fissions is just amazing. But of course, we also have our defense system because we also have white blood cells to try to defend our bodies. But still, the amount of bacteria, harmful bacteria, somehow they are immune from the white blood cells. Okay, I hope this is quite clear so far. Next, so let's start with the main core of this laboratory. Okay, the materials that will be used in this laboratory. The first one is the bacterial culture, which is E. coli, and then LB broth, and then NaOH, natrium hydroxide, and then phosphate buffered saline, and then 70% of ethanol and distilled water. Okay, and then the equipment that we will be used in this online laboratory actually cuvette, and then incubation chamber, Erlenmeyer flask, and then pipette tips, test tube, spectrophotometer, pH meter, Bunsen burner, and loop. And then now the procedure. So firstly, we have to prepare the algae broth. Okay, how to prepare the algae broth itself? First, we have to calculate the amount of media needed to grow E. coli on algae broth. Okay, we have to read specifically in the media. For instance, we need 4.2 gram in 100 milliliter of water, for instance. Then we have to do this as instructed from the bottle. Okay, and then we have to sterilize the media at 121 degrees Celsius for 15 minutes. So I know it's quite difficult to imagine how these things happen actually. Therefore, for better understanding, please check this YouTube, access to this YouTube from your own PC. And after that, you can continue to the next slide. Okay, take your time. All right, I hope that the preparation of other broth quite clear from the previous slide, also from the YouTube that you can access from your own PC. And then the next one is the preparations of bacteria. Okay, first using a sterile inoculation loop, select a single colony of E. coli from a straight plate culture and inoculate 4 milliliter liquid media in a 15 milliliter sterile culture tube. Okay, firstly we have to have this straight plate culture that consists of E. coli, and then we have to prepare 4 milliliter liquid media in 15 milliliter sterile culture tube. And then we want to incubate, we want to select um, colony from E. coli from stick plate into the 4 milliliter liquid media. And then after that, we want to incubate this in the liquid media at 37 degrees Celsius with shaking at 210 RPM overnight, approximately around 16 to 18 hours. Okay, the bacterial transfer from solid agar to liquid media should also be carried out aseptically, as we have already learned about aseptic technique, so I believe you have mastered it. And then I know for better understanding about how to transfer the bacteria from the straight plate into the liquid part, so you can also access from this video from the YouTube. And for the preparations of bacteria, you can also see from this link that shown from this presentation, from this slide. Okay, please take your time for a few minutes just to listen and then try to understand, try to imagine from the YouTube that you can access from your PC. And then you can continue to the next slide. All right, then, so this is the final step of the laboratory for today. So we would like to determine the OD600 of blank sample by putting 2 milliliter of blank per cuvette. In here, maybe it's quite confusing here. what is OD600? OD600 is an abbreviation of optical density 600. So optical density 600 in here it means it's a wavelength that we use for adjusting the nanometer, the wavelength in the spectrophotometer. Okay, and then why 600 is used? Because it's not harmful to the culture, so the bacteria will not be killed, and at the same time also this wavelength is not absorbed by the LB brood. So in here we can clearly count the amount of bacteria based on the absorbance. Okay, I hope it's quite clear for what is optical density and why we use 600 wavelength. Okay, now the second one, so we would like to inoculate the flask media with the ratio 1 to 1000 volume of the prepared E. coli culture. Okay, add 100 microliter or 0.1 milliliter of E. coli liquid culture to 100 milliliter of LB broad media. So as the first step, we already prepared the liquid media. Okay, in the first step, if you remember, and then we take this 0.1 milliliter of E. coli liquid culture to 100 milliliter LB media from the first step, the first procedure that it has been explained in the previous slide. And then we swirl the media to evenly distribute the bacteria. And then finally, we take 1 milliliter of bacterial culture into a spectrophotometer cuvette and measure the OD600. Okay, and this will be considered as 0 hour. After that, what we have to do, so we will re-incubate again the E. coli media at 37 degrees Celsius with shaking at 210 revolutions per minute and 10 points every 30 minutes. So in here it means that, okay, so let's use this cuvette. We put this 1 milliliter into a cuvette and then we put this into a spectrophotometer and we measure the OD600. Okay, the OD600, the range of the OD itself is from 0 to 1. Okay, the absorbance. Okay, and once we have the 0 time, and then we incubate again the E. coli media 30 minutes into the incubation chamber, and then we take it again, and then we measure again for the OD for every 30 minutes. Okay, at each time point, we withdraw 1 milliliter of bacterial culture and dispense into a spectrophotometer cuvette. And then wipe the cuvette clean and record the OD at 600 nanometer. And then finally, we record the result and plot a line chart in Excel. Okay, I hope it's quite clear. If it's not quite clear, then after this, we will have some discussion also in the classroom during the Google Meeting time. And then, for better understanding, I always put this video to know how to use spectrophotometer. Maybe you still get confused. You know the QFAT, but you don't know how to use. So there will be an explanations based on this link on YouTube. Okay, take your time for a few minutes and then try to listen and then try to understand the materials based on this YouTube link. Okay, and then this is the references of this laboratories for today, for today's lab. So that's all for today. I hope it's quite clear. If you have any questions, you can always ask just right after this presentation has been explained. Okay.